Maria, you are free to start speaking as you wish.
Okay, so should I start now?
Yes, go ahead.
Yeah. Okay. Hello, everyone. My name is María José Navarrete, and I'm a PhD student at UC Berkeley. And today I would like to share with you what I've learned about the role of microbial partners as architects of convergent phenotypes using tetrodotoxin in Harlequin frogs as a study system. Okay, so I think we can all agree that all of the animals depicted on this slide represent a remarkably diverse array of species whose last ancestor probably dates back to hundreds of millions of years. But they all share a very unique adaptive trait. All of these animals are protected by the same exact toxin, a potent neurotoxin called tetrodotoxin or TTX, which blocks voltage-gated sodium channels, causing paralysis and even death in organisms that ingested with a higher lethality than even cyanide. But surprisingly enough, the animals shown here are not a full representation of how widely distributed tetrodotoxin is. In fact, in this tree, you can see all of the major clades where one or more taxa have been reported to possess tetrodotoxin, encompassing a lot of organisms from terrestrial and marine systems. The current evolutionary frameworks of trait evolution cannot fully account for how widespread tetrodotoxin is because it will require a single origin of this toxin followed by multiple losses or multiple independent gains of the trait, which is difficult to believe given the distribution of tetrodotoxin across the tree of life. And this is where a new paradigm may be better at explaining the repeated appearance of tetrodotoxin in animals, which is through the association with symbiotic bacteria that can lead to the appearance of similar phenotypes even in distantly related taxa. And in fact, there is evidence coming from marine systems of tetrodotoxin producing bacteria. So here you can see all of the animals that or the groups where bacteria have been reported to produce tetrodotoxin, pointing to a similar source of this compound in terrestrial systems. But evidence was lacking from terrestrial systems until very recently. Amphibians are the only known terrestrial vertebrates to possess tetrodotoxin. And here you can see how even at this scale, there is a wide convergence in the presence of chemical defenses, particularly tetrodotoxin across different orders and also globally. And I would like to present the case of bacteria underlying the production, coming from evidence already from a species of the genus Tarica, but also present the case of how Atelopus represent an ideal system to study this at a smaller scale. So Atelopus is the only genus known with numerous species to process tetrodotoxin, which are species that you can see here in the phylogeny. And it's very interesting because they also possess not only tetrodotoxin, but also unique analogs, such as kirikitoxin and zetekitoxin, which is a hexitoxin analog, allowing us to explore convergence at a multi-species scale, but also at a multi-chemical defense scale. However, the majority of species that have been tested in this system are concentrated in Central America, leaving us with a big gap of our understanding of how widespread tetrodotoxin is in the system, but also of the evolutionary history of the trait, which furthermore prevents us from testing a symbiotic hypothesis. And here is where I would like to fill this gap by mapping the chemical defenses across Atelopus, both at presence and concentration level, as well as their geographic distribution, reconstruct the evolutionary history of toxicity in Atelopus and testing a single versus multiple origins, and finally test the symbiont hypothesis by isolating and testing for the presence of tetrodotoxin in bacteria coming from the skin of these animals. So this work integrated a lot of field work. Here in this map, you can see all of the localities I visited to collect animals for this study. And something that I would like to highlight is that many of these species are categorized as critically endangered, making this work not only extremely difficult, but also very urgent. And it led me to visit many different environments and sample species across a very wide altitudinal range, and to collaborate with local researchers back in Ecuador and Colombia. And for this, I was able to obtain 173 skin biopsies because, as I mentioned, these animals are highly endangered, so I couldn't collect whole animals, with a total of 14 Atelopus species, doubling the number of species that have been tested so far in over 50 years of research. Additionally, I was able to take advantage of an ex situ conservation program that had animals in captivity, which allowed me to test or to control for environmental variation in the samples that I tested. All of those samples were analyzed using liquid chromatography coupled with fluorescent detection, and then further confirmed using mass spectrometry by Professor Mariyotsu Yamashita in Japan. And here you can see my results. So here in the phylogeny, you can see all of the Atelopus species that have been tested, and in black are the species that were newly incorporated in this study. And in parentheses, you can see the number of positive samples over the total number of samples that I tested. So first of all, I was able to identify 7 out of the 14 species as toxic. And something very interesting that I would like to point out is that there is a wide distribution in the concentration of this compound in Atelopus. So here the X scale is logarithmic scale. So you can see that actually there are individuals that are not as toxic, but some of them are extremely toxic. And something that we also found that was very interesting is that we were able to find Hierarchical, one of the analogs, in Atelopus bombolocus, this species here that was found at 4,000 meters elevation. And this compound was previously only reported from Central America, allowing us to also explore convergence at different chemical scales. The distribution of this gradient of toxicity can also be seen here in this ancestral state reconstruction using the mean log, the log mean of the toxicity of the species. And here again, in black in the phylogeny, in circles in the map, and in the pictures, you can see all of the individuals that were incorporated in this study. You can see that the early divergent lineages are actually not as toxic, with some clades increasing or like showing a pattern of directionality of toxicity, which might be driven by interactions with local predators. Contrastingly, the Andean lineage, the sister clade to the Central America group, shows low levels of toxicity, which is something that we are still exploring, but we again see individual cases of high toxicity as it was the case of Atelopus bombolocus. Furthermore, we perform ancestral state reconstruction in binary data, and our results suggest equal probabilities of the ancestor of Atelopus being toxic and non-toxic, which suggests an environmental origin of this compound. This is something that, along with evidence of how diverse the concentration of toxicity is in the species, can point to, again, an environmental source of toxicity, potentially mediated by symbiotic bacteria rather than a strictly genetically encoded toxin production mechanism in the host. This environmental source can be further supported by our results when we compare animals collected in the wild and also animals collected in lab settings. So here you can see the results of comparing different populations for all of these species. And something very interesting is that animals that were born and raised in captivity never show the presence of toxicity, suggesting that whatever the source is not present in these controlled laboratory conditions. However, when animals were collected in the wild and were brought back to captivity, they do persist having tetrodotoxin even after years of being in captivity and on toxic free diets, which can suggest that if environmentally acquired, the toxin is not directly sequestered from food sources, which gave us again an indication of a symbiotic origin. And this is when I became a microbiologist somehow. And I got in collaboration with researchers from Universidad San Francisco de Quito. We cultured and isolated bacteria from toxic individuals, and then tested for the presence of these compounds using a commercial ELISA method that was developed for detecting tetrodotoxin in food sources because it is a major health problem, especially in Asia and Europe, as it can contaminate shellfish. And sure enough, we were able to find 9 bacteria isolates out of the 45 bacteria taxa that we tested that were positive for tetrodotoxin. Something that it is very important to mention here as well is that I also isolated bacteria from individuals that I confirmed were non-toxic, and all of those, or the samples that I was able to test for these frogs, were non-toxic as well, supporting that the idea that toxic associated bacteria are specific to toxic animals. We then sent some of these samples to further confirmation using LCMS in the UK, and we were able to confirm the presence of tetrodotoxin in some of these samples. And then we also sequenced the genomes of these bacteria to be able to identify what they are, right? So here are all of the bacteria that we were able to identify with their respective concentrations. And something really interesting is that many of these, or all of these species are known to be part of the skin microbiome of amphibians, but many of them are from genera that have been already reported in other tetrodotoxin systems, such as the genus Pseudomonas in Tarica, Rautella in pufferfish, and Staphylococcus in starfish, which can give us an idea that amphibians, as well as other marine systems, are obtaining all of these compounds from bacteria partners. So here I provided evidence for 14 new species, which 7 of those are actually toxic, increasing our understanding of how chemical defenses are distributed in the genus Atelopus, as well as showing the great variation that we see in the concentration of this phenotype in the taxa. And I was also able to provide evidence for a symbiotic origin in comparison with endogenous and dietary alternatives for the source of this compound. Further supporting the new paradigm of a symbiotic mediated convergent phenotypes that again can arise through this establishment of friendships, basically. And this finding has broader implications on many aspects of evolutionary theory, because so far complex traits such as chemical defenses have been mainly studied in the context of 2 interacting partners, prey and predators, and how their genetic mechanisms have given rise to different aspects of this trait, like toxin resistance or the physiological aspects of toxin storage and sequestration. Moreover, this also shapes our understanding of the evolution of aposematism signals and mimicry, because it is not only predators and prey, it is also bacteria that it is playing a role in the evolution of these other aspects of chemical defenses. Moreover, these patterns may also shape how we think about coevolutionary interactions and arms race dynamics occur. So, if toxicity, at least partially, is explained by symbiotic bacteria, then escalating phenotypes, as something that we've seen in Tarica and Tabnophis, with toxicity and resistance increasing as these two partners interact, might not only depend on host genetics, but also on the evolutionary and ecological
Maria,
dynamics
I'm
of host
sorry, but I'm going to have to cut
microbes.
you off. You're running over time.
Okay, sorry. And I'm done. Thank you.
All right, Jill, if you want to share your screen, anyone who has questions for Maria, I'm going to direct you to the Q&A feature. You'll be able to ask questions in text over there. But Jill, you can start presenting whenever you are ready.
All right, look good?
Yep.
Cool. All right. Hi, everyone. My name is Jill Love, and I am a recent grad of Tulane University. I'm excited to tell you more about my dissertation research linking a genomic locus for leaf-shaped phenotypic plasticity to natural selection in the wild. So first, what is plasticity and why is it important? So phenotypic plasticity is defined as the ability of one genotype to express different phenotypes in different environments. The way I like to think about it is that organisms are not plastic, their traits are. So this figure is just one example. This figure shows the plastic response of two different anole species to their walking substrate, narrow or broad. You can see that their response is a plastic change caused by their environment. So lots of organisms have plastic traits, like copepods change their phenotypes based on water temperature, desert mice change their coat color in response to ground color, monkey flowers I'm excited to tell you more about today. So, genotype by environment interactions are how selection acts on plasticity itself. And we visualize this plasticity in these graphs called reaction norms. The reaction norms I find most interesting are the ones circled here in this third panel, where we have variation in plastic response, or genotype by environment interaction. So, this variation in plasticity allows for selection to act on plasticity itself. And when does this happen? When does selection act on plasticity? So plasticity can be locally adaptive if it increases fitness by pushing a phenotype closer to a fitness peak or right to that peak. But just plasticity can just as well be maladaptive if it pushes that organism's phenotype further away from the fitness optimum. And of course, organisms are adapting to their environments, the things around them. And environment quality directly affects sessile species like plants. They can't move from this place to another. They're stuck right where they are. So environmental heterogeneity impacts the environment quality. There are a couple different ways that this takes place. So in spatial heterogeneity, we can see different distribution of resources, like in the sympatric site of Mimulus cutatus and Mimulus laciniatus. It's the same geographic location, but the water resources are very disparate between those two micro-habitat spots. There's also temporal availability of resources, such as water seen here, varying across years in the Sierra Nevada. Some years are higher, some years are lower. So heterogeneity is thought to be a key driver of plasticity evolution. This kind of makes sense because one way to respond to environmental variation is to be variable yourself. We see this in theoretical models like this one from Sam Shiner on the left, where as temporal variation increases, plasticity does as well. We also find this idea supported by empirical data, like this graph of the common frog's response to variability in pool drying. As the coefficient of variation in pool drying increases, so does plasticity. So I'm interested in this big question. What is the adaptive significance and genetic architecture of phenotypic plasticity in extreme environments? And to answer this question, I use the monkey flower Mimulus laciniatus. It is closely related to the model species Mimulus cutatus. It's a self-fertilizing, short-lived annual plant known to have low gene flow between populations. It's endemic to only the Sierra Nevada mountain range, and it grows from about 1,000 to 3,000 meters in elevation on these rocky granite outcrops. So plants regulate their lives through leaf physiology, using them for things like photosynthesis and transpiration. So there must be a reason why leaf shape mean variation and plasticity exists. So one reason in this system could be that more lobed leaves create a thinner boundary layer. So when plants get cold, leaf lobing could offer a benefit of freeze tolerance by maintaining the leaf temperature at or above freezing. And in mountainous systems, like where these monkey flowers grow, often elevation and temperature interact to create a gradient and experience photoperiod or day length. So in early spring, only the low elevations have warmed up enough for plants to grow. This happens when days are still very short early in the year. Later in the summer, those low elevations have already warmed and dried, so annual plants have stopped growing. But high elevations have only just warmed up, and plants at high elevation will experience those longer, later in the year, photoperiods as a result. So we tested whether this environmental phenomena affects monkey flower phenotypes across their altitudinal range. And in this reaction norm plot of roughly 100 genotypes from 21 different populations, we see a lot of crossing reaction norms indicating high G by E, or genetic variation in plasticity. We also find that this variation in plasticity correlates with elevation, where plants from high elevations have stronger leaf shape plasticity than those from low elevation. So now that we know this plasticity exists, how do we test for selection on plasticity in nature? I used recombinant inbred lines, or RILs, to measure selection on plasticity itself. RILs are great for this because they recombine and subsequently homogenize the genome over generations of inbreeding. So my RILs were created between one very plastic line seen in blue and one non-plastic line seen in red. After generations of inbreeding to the F8 generation, eventually genetic fixation is achieved, and each RIL has a unique homozygous combination of alleles. This creates a lot of fixed phenotypic and genetic diversity that we can use experimentally. So to measure the genotypic expression of traits in these RILs, I first grew them in long days in the greenhouse to measure their mean baseline traits. And I also grew them in both short and long days in the growth chamber to measure their plastic response to photoperiod in each genotype. This allows us to quantify each trait as a property of the genotype itself. So we captured a high amount of phenotypic diversity in the RIL population from this cross, not only in leaf shape, but in leaf lobing plasticity as well. We characterize this plasticity in three groups based on the average expression of native populations. So the first being this low elevation plasticity, where leaves became more lobed in short days. Some RILs had no plasticity at all, where the leaves didn't change between photoperiods. Some RILs have this high elevation plasticity, where the leaves become more lobed in long days. So these leaf-shaped plasticity RILs allow us to test for selection on plasticity across different environments, like low and high elevation. And I conducted a reciprocal transplant experiment in Yosemite National Park, their native habitat, to see if we can detect selection on plasticity using almost 5,000 plants. There's a little picture of me planting them all. So if I boil all of that work down to one result, it's that we find leaf-shaped plasticity is broadly adaptive across their altitudinal range. And if we group these RILs into those three categories of low elevation plasticity, no plasticity, and high elevation plasticity, we see that low elevation plasticity is adaptive at all elevations, both low and high, where high elevation plasticity also benefits only at high elevation. So now we have empirical evidence of selection on leaf-shaped plasticity. I was interested to know more about the genetic basis of this trait. And we can use the variation in RIL phenotypes to our advantage in quantitative trait locus or QTL mapping. By genotyping the RIL population, we can link phenotypes to genotypes. Essentially, we can determine what points of the genome are statistically associated with that phenotype. And this shows up as QTL peaks, where any peak over the significance threshold is a location in the genome that's associated with the measured trait. What I found when I mapped this trait is a single locus peak on chromosome 10. This peak explains almost 11% of the variance in phenotype of leaf lobing plasticity, and it has a moderate to large effect size based on Cohen's d. So when I look at the closest heterozygous marker to this peak, we find that the parental genotype of this marker significantly contributes to the real phenotype. In real genotypes heterozygous at this locus, plants with a low elevation parent's allele have a less plastic phenotype than plants with the high elevation's parent's allele. The high elevation parent was originally the one that was more plastic. So, do we see genomic signatures of selection in native habitats? Now that we have this genetic basis of this trait mapped, that's the last thing I wanted to know is, do we see a genomic signature of selection on this trait? So I took my mapping project one step further by growing the offspring from my field reciprocal transplant experiment using an evolve and resequence approach. I created 4 pools of samples, one with all of the genotypes before selection, and 3 using only the seeds of plants that survived to reproduction. So I had 2 different high elevation pools and 1 combined low elevation pool. This way we can compare allele frequencies in the before and after selection pools to see across the entire genome where is selection happening in each environment. So in these plots, we see selection happening across the genome on the x-axis, and our y-axis is the fixation index, or FST. In FST, 0 means that there's no differentiation between the before and after pools, and 1 means that there's extreme differentiation. So these points that are in red are significantly different between the before and after pools against a 99.9% empirical threshold. And we found that one of these FST peaks in a high elevation site overlaps with the same QTL responsible for leaf shape plasticity. We used a bootstrapping approach to test for the significance of this overlap using 10,000 permutations, and we found that this observed overlap was significantly greater than expected by chance, indicating that this genomic region associated with variation in leaf shape plasticity is also enriched for signatures of selection. So back to this big question. What is the adaptive significance and genetic architecture of phenotypic plasticity in extreme environments? We found a single moderate effect QTL underlying leaf-living plasticity under positive selection in a high elevation environment. And this finding is super exciting because it adds evidence that adaptive plasticity can have its own tractable genetic basis and can itself be a target of natural selection in the wild. So lastly, a huge thank you to all the people that helped with this work, especially all of the field project support. And thank you to my funding sources, and thank you to the audience for listening. With that, I'm happy to take any questions.
And we have plenty of time for questions. We got 3 minutes until our next speaker needs to begin. So if you want to ask a question live, use the raise your hand feature. Ta-da. All right, so Andrew, go ahead.
Hi, Jill. Great talk.
Thanks.
I was curious if you explored any of those other FST peaks and if anything is interesting among those loci.
Yeah, I have not yet. This is all pretty new data. This was the last chapter of my dissertation. So there is still so much more to be explored with this. And that is a great question that I hopefully will be able to answer for you in a few months.
I may as well ask myself. So I think I caught this in your slide, but I wasn't quite sure. So you had your separate pools for both of your high elevation sites post-selection, but you pooled everything together for the low elevation. Was that just because of that low survival, or was there any other reason for that?
Yeah, it was exactly that. Just really low survival. There's much stronger selection at low elevation, so we didn't have enough sample size to make different
That makes
site
sense.
pools. Yeah.
All right. So I apologize if I'm going to get your name wrong, but Jigar?
Uh, yeah, so great talk.
Thanks.
I was wondering if uh, what was the exact signature of positive selection that you discovered? I mean, I saw the FST peaks and everything, but like, uh, how did you how, like verify that it is positive selection?
Yeah, so basically we're saying it's positive selection from the field data at this point. I
Mhm.
haven't verified like on a genomic level where that lies. Because we verified that it's statistically significant at the genomic
Right.
level just based on bootstrapping.
All right. Okay. Okay. I was
All
just
right, so
wondering if
this way
like
I'm going to ask you to stop sharing your screen so that our next person can get set up. I do also want to remind both the audience members and the speakers that there are actually some questions over in the Q&A feature, so feel free to head over there to either ask or answer some questions. If you're not sure how to get there, go to the more with the three dots and choose the Q&A within there. That being said, Laura here is our next speaker, and I will let you know when to begin. It's going to be about 15 seconds. All right, you can go ahead.
Hi everyone, my name's Laura. I'm a PhD student in the Esposito Alonso lab, and I'm going to be talking to you about my thesis work, mostly focusing on predicting and verifying adaptation and maladaptation in Arabidopsis thaliana. Arabidopsis is this mighty model plant system you see here, and like many other plants, it is going to have to adapt to climate change. Arabidopsis is already adapted to many climates. Here you can see its geographic distribution, very wide geographic distribution. It's native to Eurasia, but it's been naturalized globally. And throughout its distribution, it survives in extreme and harsh environments. You can see here one of its native climates is the hot, dry Mediterranean region, and it has adaptations to do well there, but it also does well in cold, wet, and dark Northern European regions. So it has a variety of traits and genotypes that allow it to survive across these diverse locations. And scientists have studied the adaptations across space mainly through common garden experiments, where many genotypes are planted in a single location. Here you can see a collage of common garden experiments that I reviewed in an Arabidopsis Ecology and Evolution annual review paper a few years ago that showcases the diversity of common garden experiments that have been used with Arabidopsis, from the experimental design, the location, and the accessions used. But they're all unified on a similar type of question, which is there local adaptation and to what extent, and how do we quantify it? Here is a canonical example of a reciprocal transplant common garden experiment where accessions that were from Italy and Sweden were reciprocally transplanted into each of the respective locations, and fitness was measured. And it was found that there was local adaptation, that the accessions from the home environment had a higher fitness in their home climate than in the away climate. And this difference in fitness is both heritable and genetically based. And I wanted to build my PhD work off of this huge body of Arabidopsis common garden literature, but with the added element of climate change. So I wanted to build a common garden that had a climate change element in order to assess the amount and extent of adaptation and maladaptation with Arabidopsis thaliana. To do that, first I built a climate change common garden experiment when I started my PhD. I wanted to explore axes of variations in precipitation because with climate change, both the frequency and abundance of precipitation are changing globally. To do this, I built this common garden that has both rainfall addition and exclusion structures, and the control that these structures allowed us to have created a full factorial design of both frequency and abundance of rainfall, totaling to 14 precipitation treatments total. You can see those precipitation treatments represented here graphically, where at the top we have our most well-watered treatments, and at the bottom we have our highest stressed, least watered treatments. And you might think with this experimental design, we should plant a huge panel of Arabidopsis thaliana accessions and see who does well, but I wanted to take a more targeted approach. I wanted to use information from all of the previous Arabidopsis literature, as well as evolutionary theory, to create curated populations that we could push to the brink of adaptation and exponential growth, and then also maladaptation and extinction to really push the limits of this climate change common garden to see what might happen in the future. To do that, we selected our accessions with a genetically informed approach. We used two state-of-the-art methods to explore two different axes of genetic variation. First, we used a polygenic score method. I conducted a polygenic score analysis using fitness data from a previous Arabidopsis common garden experiment that was done in a hot and dry climate. We got a polygenic score, therefore, for survival in hot, dry climates for all of the 1001 genomes encapsulated in the 1001 Genomes Arabidopsis project. So each accession has its own polygenic score for survival in hot and dry conditions. I'm going to call that adaptation score hereafter. And we selected the top and bottom 10% of those accessions, planted them in our common garden to really push the two limits of very well adapted and very poorly adapted along our climate gradient. The second axis of genetic variation that we explored is mutation load. To measure mutation load, I counted the number of non-synonymous and synonymous changes in protein coding regions of the genome and created a ratio to get an estimate of the genome-wide burden of deleterious mutations to see if that had an effect on fitness. Again, each accession in the 1001 genomes has a measurement for mutation load, and we selected the top 10 and bottom 10% of accessions to, again, push the limits of adaptation and maladaptation in this experiment. Importantly, these two axes of genetic variation are not correlated. They're statistically independent enough to be considered orthogonal axes of genetic variation, so we'll treat them separately. Once I had done these analyses, I ended up selecting 245 accessions from the 1001 genomes to plan the experiment. With replication and across the 14 treatments, we ended up planting over 24,000 plants on a single day in our climate change common garden. This was a huge community science effort, and it was a great day of fun and planting for plant biologists and non-plant biologists alike. Thank you to everyone who was a part of that. Before we get into the predictions, I just want to show you that our climate change treatments worked. You can see here our precipitation treatments ranked from best to worst, and our survival and fecundity both decrease as the environment gets more stressful. But focusing now on our predictions, you can see in this figure here the same precipitation gradient, well watered to high drought stress, and our two axes of genomic variation. You can see as the environment gets more and more stressful, the predictability of the axes of genetic variation on fitness, survival increases. So populations with higher adaptation scores had higher fitness, and this was more prevalent in the high drought stress environments. And populations with high mutation loads had lower fitness, especially in the high drought stress environments. Now, this is particularly interesting because this experiment is essentially many climate change experiments that we conducted simultaneously. And we only see the predictive effects of this genetic variation when we are in the high drought stress environments. If we had made these predictions and planted them in well-watered environments, we would not have seen the effects. We're looking at cryptic genetic variation that is getting enacted under future climate change scenarios. Now, if we zoom in on the most stressful drought treatment, we can look at a model of the interaction of our two genetic variables and predictability on fitness. Here, we're zoomed in on the most extreme drought treatment. We have our X and Y axes as our two axes of genetic variation, and the points are colored by fitness in the high drought stress environment. And what this figure is intuitively telling us is that if you are an individual with a mutation load below 0.75 and an adaptation score above 0, you have a greater than 70% chance of surviving in the high drought stress environments. Now, this is more than with either of those axes of genetic variation alone. The interaction is significant, and there is synergy between these two axes of variation. Now, we learned a lot of stuff from this experiment, and I can't share it all with you now, and I'd love to talk to you more about the other questions we asked later. But the thing I want you to take away is that these dual axes of genetic variation do expose maladaptation, especially in stressful environments, and there is a positive synergy between the axes of genetic variation. But this was in a climate change common garden experiment, and we don't know if there's actually maladaptation happening with Arabidopsis in nature, which is the emphasis of the last part of my talk. In other words, can we quantify if native populations of Arabidopsis are lagging behind climate change if they have not been able to adapt to their changing climates? To test this, we took a meta-analysis framework using the previous common garden experiments I mentioned in the introduction. Here you can see a globe where points represent common garden experiments that were used in this meta-analysis, and the lines represent an accession's origin, and it is drawn to the common garden in which it was planted. Inadvertently, these common garden experiments were happening during years that there was climate change, even if they weren't intending to be a climate change experiment. You can see here climate deviations for each of these common garden sites relative to the baseline temperatures from the years 1970 to 2000. A black point on one of these lines, which each line corresponds to a single common garden, means that a common garden experiment actually occurred in that year. And you can see most of the black points are occurring in years with red bars, warm climate deviations, warmer than usual. So together, we can take all of the fitness data across these experiments to see if the populations have actually adapted to those warmer changing climates. Under a local adaptation scenario, in nature, we would expect populations to have a highest fitness in their home environmental temperature. Now, when we're thinking about reciprocal transplants, we need to visualize it a little differently. You can imagine that as this Gaussian fitness function centered around 0, and it's centered around 0 because populations will have their highest fitness when they are planted in a garden that matches the temperature of the accession's home site temperature. Under a climate change scenario, however, you might expect populations to be adapted to a previous cooler temperature. Its former environment was cooler, so the fitness peak might not be exactly at 0. In that case, we would expect the fitness peak to be left shifted, and therefore we are measuring some type of adaptation lag. The population has not yet caught up to changing climate. To see where this peak falls for Arabidopsis across all these common gardens, we updated a Gaussian fitness model. Here's the equation for a Gaussian fitness model that is represented by the black curve you see in the corner of your screen. And we updated a few parameters in this model in order to see where Arabidopsis falls globally on this x-axis. Importantly, we pulled data from the temperature of the garden site in the year the experiment was collected, the temperature of the genotype's origin site in the year that the accession was collected from there to get the difference between the garden and the origin. But we have an additional term here, that's lambda, that's adaptation lag, and that tells us how far on the x-axis the fitness curve will fall for Arabidopsis. Once we gathered all of our common garden data, we had in total data from about 42 common garden sites, spanning over 1,600 unique accessions, totaling to about 28,000 observations. We plugged these observations into this model to get a mean adaptation lag for the species. We found that there indeed was an adaptation lag for Arabidopsis thaliana. Arabidopsis from our model is adapted to approximately 1.9 degrees Celsius cooler temperatures than the temperatures in which the populations currently reside. This is in line with temperature differences that we see in the common gardens that this meta-analysis was conducted on, which on average in the past 25 years have seen an average of 1.5 degrees Celsius increase in temperature. So they've changed almost the same amount, and Arabidopsis has not caught up to the change in temperature. We're now extending this framework to other species. Despite limited data, early results suggest similarly widespread adaptation lag, but it is often greater than that we measured with Arabidopsis. Together with our climate change common garden experiment, we are suggesting that even populations that appear well adapted today are expected to experience substantial fitness loss under continued climate change. And this has devastating consequences for both population health, biodiversity, and ecosystem resilience. And with that, I would like to thank my lab, all of the Common Garden volunteers, my funding sources, and I'm happy to take your questions.
So we've got just over a minute, so we can probably afford one question live if somebody wants to raise their hand. Laura, I will ask you to stop sharing your screen just so our next person can get theirs set up.
Yep.
Da, da, da. All right, we do not look like we have a question coming in live, but we have had a few questions asked over in the Q&A, so that remains an option for people who want to ask more about this.
How'd that look?
It looks good. We're going to have you wait another 30 seconds, which I know is uncomfortably longer if we're actually silent than it seems like it would be. And all right, David, you can go ahead and get started.
All right. All right. Thank you everyone for being here. I'm David, and I'm gonna share part of my dissertation thesis on the macroevolution of hierarchical defenses in birds. I want to start by showing you this photo with different habitats in Sierra Nevada, Santa Marta, Colombia, my home country. And in places like this, and of course in a broader scale, one of the big questions that ecologists and evolutionary biologists have been interested for decades is how species adapt and disperse in these different places. Through the conference, we have seen like different ways in which organisms can like deal with this. But I'm particularly interested in how birds do that during the more early and fragile state, the reproduction. And so birds are a very good system to test that because birds reproduce in a wide variety of habitats. And also they have these very interesting strategies to deal with different environmental and ecological factors. But first, let me show you a little tour. There are some species
So,
that...
David, your slides are not advancing.
Ooh, let me... How can I share that? Stop sharing. Now sharing again, I guess.
It's showing your desktop.
Okay, I'm gonna share again. It's now changing?
Yes, you're not on your title slide anymore. Good. We
Okay.
see a video.
Great. So I was telling you about this saltator that built this nest, cup nest in the vegetation, but other species have these more creative materials, and they use fungi and spider web to like attach underneath the leaves their nest. There are other species that have these more complex nests with hidden entrance, while others like use cavities to hide their nests, or even just lay their eggs on the ground. In all cases, the main goal is to protect the embryo from like these rough environmental conditions, and of course try to like avoid predation. And so all these multiple layers of defense are protecting the embryo, like consisting like a parental behavior, nest site and type, and also eggshells that protect finally the embryo. For today's talk, and these are protecting from predation and also environmental factors like damage, humidity, temperature, radiation, and infection. And for today's talk, I'm gonna focus on nest and eggs. I'm gonna talk a little bit about nesting ecology and how that might be interacting with eggshell structure, and then show you the diversity in eggshells that we have been finding, the diverse nests that are associated with that, and hopefully at the end, I would like to convince you that studying this system is very interesting, not only from the natural history perspective, but also to ask evolutionary questions. I would like to start then with this major transition. And this major transition is nesting from the ground. And this is an ancestral state that tracks back to dinosaurs, and to build elevated nests on branches and cavities. And one of the advantages to having this different nests or to elevate their nests is like the risk of infection is lower. And I will argue that this single transition unlock different reproductive strategies and novelties in these phenotype and reproductive traits. But first, let's take a close look to the eggshell and what I mean by an eggshell. Just remember the egg that probably you had for breakfast. And if we take a close look, there are like different layers that are protecting the embryo. For today, we are going to talk in the most external layer. This is the cuticle. And the cuticle in these scanning electron microscopes or SEM images, I'm showing you how the cuticle looks like in different vertebrates. This cuticle is protecting the eggshell, is the first line of defense in all these lineages. And especially in birds, since we have so many diversity in terms of size, shapes, colors, it has been suggested that it's highly variable. To study fine scale reproductive traits, you have to go out and do nest searching. And I spent a lot of time doing this. We even like wrote a methods to help other people to find nests, to ask this kind of questions, but also go to the museum and take samples to then extract different metrics of what you are describing in your data. In this case, I took several techniques, imaging techniques, but I'm going to focus mostly in the scanning electron microscopes. Our sampling in the fieldwork was mostly focused on Colombia and Peru, from the Amazon to the Andes, but thanks to the collections, we expand our samples like geographically and also taxonomically. And this is how it looks. After you find an S, you collect the samples and you took the images. Here again is a cross section of the eggshell, and in orange you can see the cuticle. And this is a photo from the top, and this is how a cuticle is you can see a cuticle in the paleognates or in the chicken nest. In contrast, there are a lot of other groups in which like the cuticle is absent, and this is how you see that in the right, the eggshell that with these pores and vesicles are exposed. And this is our first results for all the species included in our analysis, which is a nice cover of like around 95% of the species. We took data of nest location and cuticle absence and presence, and this is what we found. In this is a recent phylogenetic tree, and we map our traits here, but the main message is that we found 5 different events, independent events of loss of cuticle. In the nightjards, the pigeons, some aquatics, marine species, quetzals and trogons, and parrots, and all the recent radiation of the passerines. With this data, we can start to ask about evolutionary transitional pathways from this ancestral state with ground nesters, with eggs with cuticle, to species that have elevated nests with cuticle, or species that are still nesting on the ground with no cuticle. And to arrive to this last condition with elevated nests and no cuticle, there are like 2 main paths. First, they can still like nest on the ground and then lose their cuticle, or they can elevate their nest and then lose the cuticle. And what we found is that the arrows indicate the most likely transitions is that the birds in general are first expanding their nest sites and then losing the cuticle. But I have been telling you about eggshell diversity and what that means in terms of the cuticle. In this dimension, when they are expanding their nests, we found these different morphologies and quantifiable differences in shape and also chemical compounds that allow us to describe these 4 base cuticle types. And of course, we have the cuticles of these open surfaces. If we then further ask about the transitions in these different types of cuticles, we can start with the ancestral cape eggshell. That's the same one that we have in our chicken eggs. And one of the paths produce reticulated eggshells, very robust. And then in this in this next transition, there are these vesicular cuticles that are mostly found in aquatic species. There are also another type of cuticle, the occluded, and seems to be more versatile because it protects from like the small hummingbirds to the huge RP eagle nest. And then from there, you have this open surface in this species. But the next question here will be, why to lose the cuticle if it's so protective? Well, the idea here is like after you reduce that risk of infection, there is an endless open of possibilities with like traits in the eggs in terms of their color, and the different spottings in the eggshells. We have several couple of other working hypotheses. We also think about the water balance, and we have a recent paper on that. And also you can think like after losing the cuticle, there are like some mechanical properties that change. And we are also now exploring the strong breakable properties of the eggshell. But what about the nest? And in the last part, I'm gonna tell you, like, and from our tour, you probably are thinking that elevated nests are an oversimplification of the nest strategies. And even though in some groups the nest shows a strong phylogenetic signal, meaning that order level, pretty much all the species in one given order present very similar nesting characteristics. In passerines, and especially in flycatchers, there are this huge diversity in how they build an elevated nest, from a very complex cups and domes. And we were exploring all these ideas in the clade that includes the most species family in the world, the flycatchers. And we found that shifts in nest types are associated with ecological transitions during lineage diversification. We described the different patterns of evolution of all these different nest architectures. But if you want to know more, I invite you to check this super interesting issue on the evolutionary ecology of nests across taxa approach. To wrap up, I would like, we saw how these like major nesting transitions have something to do with the evolution of the eggshell structure, and then now how that went back to modify nest in kind of like this functional continuum in which like different components are related with the transitions, and they open the gate for like new innovations in the other traits related. All this to protect from all these different factors and many more, like parasitism, signaling, to really like approach this evolving network and to allow species to colonize and thrive in different habitats. Going forward, I would like to explore the genetic basis of, for instance, the cuticle. There are like some candidate genes that have been explored, and also the chemical composition. And I'm particularly interested in the mechanical properties and the 3D models of the nest. And that's something that I will continue doing during my dissertation and like in the next steps. All this to have this integrative framework to study the evolution of this hierarchical line of defense. To conclude, by linking extended phenotypes like nests with intrinsic reproductive traits like eggshell structure, we can better understand how multiple defense co-evolve to mediate environmental interactions and promote ecological diversification across birds. I would like to thank all the people involved in the nest searching component of these projects. It really takes a village to find nest. The museum collections that have this calcified treasure there for us to do research, funding in my department, and also funding from like collaborators and my advisor to like pursue all these questions. Thank you.
All right, so we've got a minute and a half, so we probably have time for 1 or 2 questions if anyone wants to raise their hand. I guess if no one else has one to begin with, you do have this tremendous diversity in the sizes of your eggs. Have you noticed any sort of correlate between how large the egg is and how thick the cuticle is?
There is a strong correlation with that. There are like in X-rays, there are strong allometric correlation. But in terms of the cuticle, it really depends on the type of cuticle you have and each species have. And for instance, in some of the species, you might expect a huge cuticle, like very thickness of the whole eggshell on the cuticle. But then, for instance, in hummingbirds, that you have a very small egg, you have one of the biggest relationships, like one of the less thick, no, one of the thicker eggshells corrected by size. So it really depends on the clades and like other aspects of the cuticle.
All right. So we have 15 seconds left. I'm sorry, Maria. It looks like we're not going to have the option for you to ask your question live. I'm going to make you not a co-host so you can type it in the Q&A. And then Hannah, you are free to begin whenever you wish.
Probably helps if I unmute first. There we go. You can see my slides okay?
Yes, we can.
Perfect. Okay, hello everyone. Today I'm going to be talking to you about how flowers evolve in response to pollinator decline and what that evolution can mean for future generations of plants and their pollinators. So flowers are highly variable in terms of size, shape, color, and even smell. And the reason for this wide range of diversity in floral forms is pollination. Interactions between plants and their pollinators has been driving the diversification of floral forms through millions of years of evolution. But as I'm sure you're all aware, many insect pollinators have been experiencing declines in abundance. Now, from the perspective of a plant, this is a really big problem because the vast majority of flowering plants rely on pollinators for reproduction. So if plant populations are going to persist, they have to adapt to declines in pollinator abundance. Now, one of the ways that plants can adapt to pollinator decline is through the evolution of floral traits to facilitate self-pollination. This can include traits like smaller flowers, which can allow plants to conserve resources that would have otherwise been wasted on trying to attract unreliable pollinators. It could also include reduced spatial separation between the anthers, that's the male pollen-producing organs, and the stigmas, the female pollen-receiving structures. This would increase the probability that self-pollen flows between the male and female structures when there's a single flower without any external assistance. Now, although increased selfing could help plants maintain at least some seed production, it could also lead to longer-term consequences for plants, which I'm going to be calling the worst-case scenario number 1, as well as longer-term consequences for pollinators, which I'll be referring to as the worst-case scenario number 2. If pollinators are extirpated, then the evolution of selfing would allow plants to continue making at least some seeds, whereas plants that are solely reliant on outcrossing would experience reproductive failure. Similarly, if pollinators remain present at low abundance, plants that have evolved increased selfing might have a fitness advantage over outcrossing plants, because selfing could potentially supplement any outcross seed production. However, if pollinator abundance increases, then we get that worst case scenario number 1, where plants that have evolved to self could experience a fitness cost. This is because selfing flowers are typically less attractive, so they could experience reduced visitation rates, and increased rates of selfing means there are fewer ovules available for fertilization by outcrossed pollen. Additionally, because the evolution of selfing could include reductions in the quantity or quality of rewards like nectar and pollen, this could mean that the evolution of selfing could result in decreased food availability for pollinators, which could exacerbate further pollinator decline. So this is our worst case scenario number 2. So given these two worst case scenarios, it's important that we ask not only how will plants evolve in response to pollinator decline, but also what the longer term ecological consequences of evolution in response to pollinator decline will be. To address this broader question, I used an experimental evolution study to answer 3 research questions. First, does the evolution in response to pollinator decline lead to changes in floral traits that facilitate selfing? 2, does it cause fitness consequences when pollinator abundance changes? And finally, could it trigger a reward-based feedback loop that exacerbates further pollinator decline? To answer these questions, I used Mimulus guttatus, which is a wildflower native to the western regions of North America. The species is primarily pollinated by bumblebees, but it can also readily self-pollinate when pollinators are scarce. The species produces very minimal quantities of nectar, so pollen is the primary reward in M. guttatus. Now, although there are both perennial and annual populations of this species, the seeds used for this study originated from an annual population on Iron Mountain in Oregon. I raised outcross seeds to flowering in a pollinator-free greenhouse, then randomly assigned plants to either a high or low pollination treatment. These pollination treatments occurred simultaneously, but in separate flight cages housed inside a single growth chamber, which you see on the right here. Plants in the high pollination treatment receive visitation from 12 to 16 bombus impatiens workers for each day for 9 days, and plants in the low pollination treatment receive visitation from no more than 3 bees per day for 9 days. I then bulk collected the seeds from each population, reared those seeds in a greenhouse, and subjected them to the same pollination treatments as the parental plants until each population had experienced 3 generations of pollination treatments. I then repeated this entire process for a total of 3 replicate populations evolved under each pollination level. To answer my first question, I grew 4th generation plants in pollinator-free common gardens. I measured flower size and anther stigma distance in 5 flowers per plant, and used mixed effect models to test whether floral traits that facilitate selfing evolved in response to low pollination treatments. What I expected to see for both flower size and anther stigma distance was that mean floral traits across populations exposed to high pollinator abundance, which is represented here by the white circles, would not really exhibit much change over time relative to that original seed source, which is represented by the black circle. But for populations exposed to low pollinator abundance, which is represented by the gray circles, mean flower size and mean anther stigma distance across populations would decrease over generations and ultimately be significantly lower in the 4th generation common gardens. However, flower size did not meet this expectation. Although mean flower size in the low evolved populations was consistently smaller than the high evolved populations, there was no consistent decline across generations and no significant difference in the 4th generation common gardens. In contrast to this, though, anther stigma distance was very well aligned with the predictions. The low evolved populations showed steady decreases in anther stigma distance across generations and was significantly lower than the high evolved populations in the 4th generation common gardens. Now, because reduced anther stigma distance can directly facilitate autonomous selfing, this leads back to my second research question, which was, does the evolution of selfing have fitness consequences when pollinator abundance changes? I quantified seed production in 4th generation common gardens that were exposed to either pollinator-free conditions, low pollinator abundance, or high pollinator abundance. And because the high and low gardens were paired temporally in a growth chamber and the pollinator-free gardens remained in a greenhouse, I used one mixed effect model for the pollinator-free gardens and a separate second model for the high and low gardens. What I found was when pollinators were absent, plants that were evolved under low pollinator abundance, which is shown here on the right in gray, produced significantly more seeds than plants evolved under high pollinator abundance, which is shown here on the left in white. Now, although a separate experiment I conducted for a different chapter of my thesis found a different result, which I can discuss in more detail later if anyone's curious, these data show that plants evolved under low pollinator abundance can have a fitness advantage when pollinators are extirpated. In the low pollination gardens, the low evolved populations, which is again shown in gray, produce more seeds than the high evolved populations, shown in white, although I'll note that this difference was not actually statistically significant. When I compared seed production in the high pollination gardens, which you see here on the right of the x-axis, there was no difference in fitness between plants evolved under high versus low pollinator abundance. So these results show that even if plants evolve to self in response to pollinator decline, does not necessarily result in a fitness cost when pollinator abundance increases. To test my final research question, which was whether evolution in response to pollinator decline could trigger a reward-based feedback loop, I measured the quantity and quality of pollen produced by evolved populations grown in pollinator-free common gardens. I used a Coulter counter to quantify the mean total number of pollen grains produced as my measure of pollen quantity, and the total number of viable pollen grains produced as my measure of pollen quality. What I found was there was no difference at all in the total number of pollen grains produced by plants evolved under high versus low pollinator abundance. There was also no difference in the number of viable pollen grains produced. Now, I realize you might be wondering if there's perhaps a mistake on this slide, because these two graphs look remarkably similar, but I can assure you these are actually different graphs. The reason why these graphs look so similar is because the vast majority of pollen grains produced by all of the plants, regardless of their evolutionary history, was of very high quality. So around 96% of all pollen grains were viable. So to quickly recap those three research questions, yes, evolution in response to pollinator decline can cause changes in floral traits that facilitate selfing. Specifically, it can cause reduced anther stigma distance. Can also provide reproductive assurance when pollinators are absent or rare without imposing a fitness cost when pollinator abundance increases, but it does not necessarily trigger a feedback loop that exacerbates further pollinator decline because evolution in response to pollinator decline did not cause a decrease in the quantity or quality of nutritional rewards. So in terms of a bigger picture, specifically those two worst case scenarios I presented earlier, where in scenario 1, pollinator abundance improves, but this ultimately leads to a fitness cost for plants, or scenario 2, where plants adapt to pollinator decline, but those adaptations ultimately make life harder for pollinators. I found no evidence for either of these two scenarios. Instead, what I really found was the best possible scenario, because I found that the evolution of selfing in response to pollinator decline could not only help plant populations persist even as pollinator abundance changes, it could also potentially help support pollinator recovery through the maintenance of high-quality nutritional resources. So if you only remember one thing from my talk today, what I hope you remember is that just because a worst-case scenario is possible, that's not a guarantee that the worst will actually happen. So I want to thank all of the wonderful, amazing people you see on this slide that helped make this work possible, and all of you for coming to my talk today. And I would be happy to answer any questions if there's time left.
And there is plenty of time left. We have 3.5 minutes until our next speaker begins. So
Ooh.
anyone who wants to ask a question, go ahead and raise your hand. Okay. All right, well, with no one having done so yet, I will take advantage of asking the first question. Like, what are you most excited about doing next beyond the work you've shown already?
Oh, what I really want to do next is see if evolving in response to pollinator decline kind of makes it easier to adapt to drought. Because selfing is actually a drought avoidance strategy for these particular plants. So I really, really want to look at, survival, germination rates, maybe even like clonal reproduction under drought.
All right, so Benjamin, you can unmute yourself and ask your question.
Yeah, great talk. I was curious about the difference in the results you saw with respect to floral size as well as the anther stigma distance, and whether you thought, given the different explanations you gave for why that might take place under a low pollinator environment, whether you thought you might see different results if, say, the abiotic stressors were different or something like resource availability, things along those lines. Do you think you might see flower change in other contexts, or do you think that it's less sensitive to contexts, maybe?
It would definitely depend on what the stressor is. Drought would be a really big one. I have another chapter where we did a field study simulating pollinator decline using bridal veil bags, and we also implemented a drought. And in that case, what we actually found was selection for mixed mating. So drought actually drove selection for bigger flowers and reduced anther stigma distance.
So I am curious, how much do you expect there to be essentially interactions between different forms of environmental resistances. So I don't know as much about flowers. I can tell you in the bacterial world, we often see correlations between drought resistance and salt tolerance because things get saltier as everything dries out.
Oh, I hadn't thought of it that way. Yeah, I mean, the tricky thing with pollinator decline and using other sort of environmental variables, it's also impacting the pollinators themselves, not just the plants. Yeah, that would be really interesting.
Just a
I'd
thing
have
to
to
think
think
about.
about that. Yeah, ideas for future experiments.
All right, so we're down to about 30 seconds left, so we're going to have Sylvia, our last speaker, go ahead and start sharing your screen. As always, any other questions people have, feel free to head over to the Q&A and add in some questions over there. Right, so we're just
Does this
going to
look
wait.
normal?
Yep, things do look normal.
Okay.
Well, I mean, as much as very, very young mice ever look normal.
Fair.
And you can begin whenever you wish.
Okay. Well, first, congrats, everybody. We've all made it to the end. I'll try my best to bring us home. So my name's Sylvia. I'm in Michael Nachman's lab at UC Berkeley, and today I'm going to be talking about the genetic and environmental basis of adaptive tail length in house mice. So, one of the most fundamental goals in evolution is to understand the genetic basis of adaptive phenotypes. And we know a lot about this process for simple traits that are governed by just a few loci of large effect. But the reality is that most evolution is taking place through complex phenotypes. And they're complex for two reasons. They're controlled by many loci across the genome, but also by potentially strong environmental influences. And today I'm going to dissect both the genetic and environmental components of a classic example of adaptive variation. So, Allen's rule is perhaps the best documented ecogeographic pattern in mammals. And this is the observation that animals that are closer to the equator have longer extremities. And we think of these as these phenotypes as thermoregulatory adaptations because having shorter extremities in a cold environment is going to help you retain heat, and then vice versa in a warm environment. So I don't study these phenotypes in rabbits as shown here. I use a more experimentally tractable system, and that's house mice. So house mice are found across a wide variety of habitats. And I specifically focus on two inbred lab strains that were originally collected from a warm tropical environment in Manaus, Brazil, and a cold temperate environment in upstate New York. And in my talk today, I'm first going to ask one, do house mice conform to Allen's rule? And second, what's the genetic basis of Allen's rule? And then lastly, I'm going to investigate the effects of environmental variation on these phenotypes. And today I'm just going to focus in on tail length for our focal trait. So one of the first things we did to address this initial question was to measure tail length in lab-reared New York and Brazil mice. And we find that, yes, indeed, these mice do conform to Allen's rule. There wouldn't really be a talk otherwise, as tail length is longer in our warm-adapted Brazil mice. And next, I use cleared and stained specimen to show that this is due to both a difference in the number of tail vertebrae and the length of those vertebrae. And lastly, we wanted to test the function of this tail length variation. So we use thermal imaging to show that New York mice with their short tails are able to retain more heat in their appendages during a cold stress. And that's what I'm showing you in this graph here. So now that we've set the groundwork for Allen's rule in these mice, I want to talk about the genetic basis of this trait of tail length. And for this, I used a QTL mapping approach and mapped total tail length as well as tail vertebrae number and tail vertebrae length. So here I am showing you the regions of the genome that showed significant association with total tail length. And if we compare that to our two other tail traits of interest, we do see that there are multiple overlaps between our different tail phenotypes. But beyond understanding just the genetic architecture of these different traits, if our ultimate goal is to isolate the actual genes that are controlling this phenotypic variation, The truth is that these regions of association we get are still pretty large and they contain hundreds of genes. But there are several things we can do to try to narrow this down. And for this part, I'm just going to focus on one peak on chromosome 4, and that's one that's shared between total tail length and tail vertebra number. So first, I gathered gene expression data in the tail of New York and Brazil mice to focus on just differentially expressed genes within our QTL window. And next, we can overlay genes that have a noted skeletal phenotype in the mouse genome informatics database. A great thing about working in a model organism. And lastly, I gathered allele-specific expression data from F1 hybrids that allows us to isolate cis-regulated genes. So here I overlap genes that had significant cis regulation, as well as those that we couldn't test for allele-specific expression, to retain as many candidates as we could. So from all of these different overlaps, we're left with just 6 potential candidates. And the gene out of these 6 that was most differentially expressed was this one called Lin28A. And this was really exciting to see because this gene is an RNA binding protein that acts to increase tail vertebrae number in mice, which you can see in this overexpression mutant in the top panel here. And that matches up with Brazeau mice having higher expression of this gene. And we know that Brazeau mice have evolved to have more tail vertebrae. And this chromosome 4 peak is the one that also maps to tail vertebrae number in our QTL. But up until now, all the data I've shared is from mice that have been bred in the lab for many, many generations under common garden conditions. So we always assumed that all the phenotypic variation that we were investigating reflected solely genetic differences between these mice. But in reality, these common lab conditions can't control for any potential non-genetic parental influence on phenotype. And for placental mammals like house mice, we know that the ultimate phenotype can be greatly influenced by both the prenatal and the postnatal environment, which is primarily provided by the mother. And these influences are deemed maternal effects. So I want to take the rest of my time today to investigate the environmental effects that are governing Allen's rule, with a particular focus on the role of maternal effects. So to study this, I cross-fostered New York and Brazil litters within 24 hours of birth, and then measured tail length every 4 days from birth until weaning, and then also did these measurements for non-cross-fostered litters. So here I'm showing you tail growth during nursing just for our pure line mice, which of course is higher in Brazil. But if we overlay our cross-fostered pups in the lighter shades and then zoom in on our final weigh-in here, we found something pretty surprising, and perhaps this is my favorite result of my whole PhD. So we saw that all pups reared by Brazil moms have long tails similar to regular Brazil mice, regardless of what strain they are. And then vice versa for pups reared by New York moms. So we can conclude from that there's actually a really strong maternal effect on tail length, and it's in the direction of adaptive divergence that we see in these mice. And then if we measure these mice into adulthood, we see that the maternal effect is dampened, but it's still present at 9 weeks. And ultimately, it's explaining about 50% of the difference we see in tail length between pure line New York and Brazil mice. And I said that this result is surprising because we assumed that maternal effects would be acting through mechanisms that might influence overall growth, not something that would just be influencing tails. Because I'll quickly note that we measured body weight as well in this experiment, and there's not a lasting maternal effect there. But importantly, tail length and bone growth more generally is known to be incredibly plastic, but particularly to different temperatures, with tails growing longer when animals are in warmer conditions. So to test this in the maternal context, I started measuring the temperatures of New York and Brazil nests throughout nursing. And what I found is that nests that were built by a Brazil mom were much warmer throughout nursing, which aligns with that hypothesis. And when I thought about what could be leading to this difference in temperature, I thought the most likely cause would be a difference in nesting behavior, because Brazil and New York mothers in the lab do build starkly different nests. So I used a rubric to quantify this and found that, yes, indeed, they do build different nests. New York mothers build these open and exposed nests, whereas Brazil mothers build these more enclosed nests. So we think that this connection between divergent nesting behavior and differences in nest temperature provide us with a potential direct link between the ambient temperature that's controlled by the mom and the ultimate tail length of the pup that she's rearing. So to round out this section, I now want to talk about how the maternal effect is reflected at the skeletal level. And specifically, I mean, is the maternal environment influencing the number of tail vertebrae, the length of those vertebrae, or some combination of the two? So first, I'll talk about tail vertebrae number. We found that Brazil and Brazil cross-fostered mice have about 28 caudal vertebrae, whereas New York and New York cross-fostered have about 26. So the fact that our pure line and cross-fostered individuals don't differ points to this likely being an evolved, genetically determined difference. But for tail vertebrae length, we found that pups reared by Brazil moms had longer individual vertebra than pups reared by New York. So this points to this likely being a plastic, maternally determined difference. So through this analysis, I was able to see which skeletal mechanism is conferring our maternal effect, and also which elements of this phenotype have a strong genetic basis. And then to finish up this last section, I want to dive a level deeper and think about the molecular basis of this maternal effect. Sorry, I lost my voice like 2 days ago. So to do this, I gathered RNA-seq data in the developing tail at 10 days of age from our pure line and cross-fostered pups. And I want to start by just showing you the PCA of all of our samples. So the first thing we see is that the main axis of variation separates samples out genetically. So all Brazil pups on the left, all New York on the right. But our second axis of variation separates them out by maternal type. So everyone raised by New York on the top and everyone raised by Brazil on the bottom. And I want to note that maternal type is actually a stronger predictor of expression than sex. So therefore, it's a really strong mediator of overall tail gene expression variation. But beyond describing things like those overall expression patterns, we wanted to try to pinpoint the gene expression differences that were actually governing our maternal effect on tail growth. So one of the first things I had to do to address that was group genes by whether they had patterns indicative of genetic control or maternal control. And I was actually mostly interested in our maternal set of genes here. So I took this set and performed weighted gene co-expression network analysis, or WGCNA, on this whole gene set. And we pulled out this module where expression patterns strongly match maternal status. And here I'm showing you just the hub genes of that module. And our labeled genes here with the asterisks are actually those genes that have a noted skeletal phenotype in the mouse genome informatics database. And I found that our hub genes are actually enriched for these skeletal phenotype genes as compared to just background gene expression. So that suggests that this module here is likely involved in mediating that maternal effect on tail growth that we're really interested in. Okay, so while there are multiple interesting genes in this group involved in things such as canonical skeletal growth pathways, I'll highlight just one, and that's leptin. Makes me emotional. So while it's mostly studied for its role in weight gain, leptin is actually one of the primary candidates for temperature-induced bone growth. So it's known that leptin is upregulated in warm temperatures, and that's known to increase levels of important growth factors and stimulate bone growth plate activity, which can all lead to bone elongation as a function of temperature increase. And this matches what we see in our expression data of higher leptin in the mice that are raised by Brazil moms, so the pups that are in that warm nest. And I want to end here with an overall model for tail length variation, which includes some loci I can highlight today. But first, I'll quickly point out that we know from previous work in Brazil and New York mice in the lab that narrow sense heritability for tail length is about 45%. And we saw earlier that the maternal contribution to tail length at adulthood is about 50%. So when it comes to tail length variation in the lab, we're actually able to account for almost 100% of the phenotypic variation we see. And we've tried to understand here how both those genetic and environmental causes are mediated at the molecular level. So zooming all the way out, as I mentioned at the beginning, almost all of adaptive evolution involves complex traits. And we have very few examples where we're actually able to understand the genetic and environmental mediators there. And I hope today you can see that we've tried to do some of that. And also, I hope that these data highlight the intrigue that comes from investigating multiple levels of biological influence in order to understand how phenotypic variation is actually generated and maintained. And I see the red no, so I'll say thank you and ask if there's any questions.
All right, so that is the end here of our third session for the Hamilton Awards. We now have the point where the committee has to make some very difficult decisions, and I do not envy them for that. We are going to have a short break before the next session start up, but before we leave, do want to thank all of our presenters for a very interesting series of student talks.